Hi everyone! Tonight's video is on organelles, which are the small parts inside of eukaryotic cells that carry out specific functions for the cell. This video is designed to give you a big picture look at how all the parts of a cell work together to allow a cell to perform its necessary functions. This video is a very different style than you've been listening to in recent weeks. There's no expectation that you're going to be taking notes. We simply want you to see from a big picture how all of these organelles can work together to perform functions for the cell before you start reading about the individual specific details of each organelle in your textbook. As you're reading in your textbook, you're going to be filling in an organizer that details the structure and the shape and whether or not each organelle is surrounded by a membrane, what is its function, what cell type is it found in? Are they found in just animals, just plants, or in both? But tonight's video is more of a general big picture look at how these organelles work together. The process we're going to be going through is the synthesis and export of a protein that is destined for outside the cell. Cells make many proteins that stay inside the cell, but they also need to make proteins that get exported from the cell. These proteins can have a variety of functions, such as fighting a bacteria that's trying to infect it, or perhaps forming the structure that is outside the cell that allows the cell to maintain its individual organization. Or perhaps this protein is a signal to another cell so that the cells can communicate for each other. The reason we have chosen this process to go through is that it takes us through many of the organelles that are pictured in the diagram in front of you. So starting with our cell that's in front of you, all cells are surrounded by what's called a plasma membrane. It can also be called a cell membrane. This is a phospholipid bilayer. Inside of the plasma membrane is the cytoplasm. The cytoplasm is an aqueous solution of salts and proteins and all of the organelles that you see listed here on this cell. We are going to start our journey of how a cell makes and secretes a protein in the nucleus, which is in the center of your diagram here in purple. Let's take a closer look at the nucleus. The diagram on your left-hand side here shows an isolated nucleus. The nucleus is surrounded by a membrane, and that membrane is often called the nuclear envelope. This nuclear envelope is in fact a double bilayer of phospholipids. There are two membranes in a nuclear envelope. That means there's a total of 4 layers of phospholipids. On the inside of the nucleus is the DNA. This is a nucleic acid that has the code for how to make all the proteins the cell needs to make. This DNA never leaves the nucleus, it's too valuable. The problem is that the information that's in the code in the DNA has to get to the organelles in the cytoplasm in order for the organelles to build the proteins. In order for this to happen, there is a structure in the nuclear envelope called nuclear pores. These are small perforations in the nuclear envelope that allow for regulated entry and exit. I say regulated because it's not just an open door. Only specific things can get in and out of the nucleus. Attached to and continuous with this nuclear membrane or nuclear envelope is an organelle called the RER, which stands for rough endoplasmic reticulum. We're going to cover this in more detail in just a few minutes. Let's look at the diagram on the right-hand side of your screen that is just a little bit more of a cartoon of what's going on here in the nucleus and the cytoplasm of a cell. So the nucleus is again shown in purple, and inside the nucleus is our double-stranded DNA, which has the code again to make all the proteins necessary for the cell. This DNA never leaves the nucleus, but the information in the DNA does need to leave the nucleus. So the cell makes a copy of the DNA in the form of another nucleic acid called RNA. The process of making a copy is called transcription, and the specific RNA that is made that has the code for making proteins is called mRNA. The M stands for messenger. This is a messenger RNA. This messenger RNA can leave the nucleus and go out to the cytoplasm. If the messenger RNA gets chewed up by enzymes, it's not a problem. It's just a copy of the DNA. We can always make another one. So this mRNA is going to leave the nucleus through a nuclear pore and go out to the cytoplasm. As soon as it's in the cytoplasm, another organelle called a ribosome is going to clip on to this mRNA. So in this diagram here, we have the mRNA and we have a ribosome. Ribosomes have two subunits, and they are generally drawn just as this picture is shown here, kind of like a snowman, with one larger subunit and one smaller subunit. The job of the ribosome is to read the code in the mRNA and use this information to assemble the amino acids into a protein. So here is our growing protein of amino acids. I'm going to label those AA there for amino acids. And the ribosome is reading the code in the mRNA and using that to put the specific amino acids in the right order, building our protein. But we're building a protein that's going to be exported from the cell. So at some point in the mRNA, the ribosome is going to come to a stop sequence or a little code that I've indicated here in blue that says, whoa, wait a minute, this protein needs to be exported from the cell. Because of this one needs to be exported from the cell, the ribosome is going to bring the mRNA and the growing protein to another organelle called the RER, or the rough endoplasmic reticulum. The ribosome is then going to actually dock on the RER. Each of these pink dots on this RER is a ribosome. This rough endoplasmic reticulum is just a series of membrane sacs that I said is continuous with the nuclear membrane, and it has all of these ribosomes docked and embedded in the membrane. Each one of these is making a protein that is destined to be secreted from the cell. Once the ribosome docks on the RER, it is going to continue making its protein, and it's going to insert this protein into the inside of the membrane sacs that comprise the rough endoplasmic reticulum. Once the ribosome is completed with making its protein, it's read all of the instructions in the mRNA, the ribosome will then disengage from the RER, and the mRNA will also leave the RER. But our protein is still in the inside of the RER. The job of the RER is to modify our protein by putting sugar units on the very ends of the protein, making this protein a glycoprotein. Once the RER has made the protein into a glycoprotein, it is then ready for its next step in the journey. In this next diagram, you can see our glycoprotein here at the end of the RER. The RER is going to pinch off a vesicle, which is just a membrane little bubble here with our glycoprotein inside. This vesicle is going to take the protein to its next destination in the cell. But this transport vesicle isn't just going to float around in the cell until it finds its right place. It's actually going to move along a structure called a microtubule. Microtubules are part of the cytoskeleton. They're made of protein. They don't have a membrane around them. And one of their functions is to function as a train track, a track for vesicle movement. So our transport vesicle is going to move along this microtubule until it reaches another membrane stack called a Golgi apparatus. It can also be called a Golgi body, or it can simply be abbreviated the Golgi. The Golgi apparatus is this stack of membranes that looks a little bit like a stack of pancakes. It's generally fairly easy to identify in diagrams because it has this very characteristic structure. Once the Golgi has received the transport vesicle, the protein is then going to go inside the interior of the Golgi apparatus, and it's going to move along the stacks of the Golgi apparatus, going from the near end to this more distal end of the Golgi apparatus. While it's traveling through this stack of membranes, it's further modified. It could be cut into shorter pieces, it could have functional groups added to it, it could have lipids added to it. A variety of modifications happen to our protein in the Golgi apparatus. We think of the Golgi apparatus as FedEx because it's involved in packaging and modification of proteins so that they're ready to go to their final destination. Once the protein is fully modified and has reached the distal end of the Golgi apparatus, another vesicle is going to pinch off. Again, this will be a membrane-surrounded vesicle, but in this case, it's called a secretory vesicle because it is now ready to secrete the protein to the outside of the cell. The secretory vesicle will also move along a microtubule until it fuses with the plasma membrane. Once it is fused with the plasma membrane, the contents of the vesicle are released to the outside of the cell. And we can now look at this original diagram of our cell and see that we have in fact covered a tremendous number of these organelles when talking about the process of secreting a protein from a cell. We started with the nucleus, and then we moved on to ribosomes and the rough endoplasmic reticulum. We talked about microtubules being the train tracks. We talked about the Golgi apparatus. We talked about vesicles and cytoplasms. We also talked about the role of the nuclear pore in getting information out of the nucleus. Hopefully this overview is going to help you when you begin reading about the specific individual organelles in your textbook, and when you get reading about more detail, you will be able to look back and see how they all fit into the organization of the cell. That will be all for tonight.